Muy buenos días, bienvenidos al ciclo de conferencias del Instituto de Genética. Es un placer para nosotros tener a nuestro invitado de hoy, el profesor Anthony de Tomaso, más conocido como Tony de Tomaso. Tony va a hablar sobre la evolución del sistema inmune adaptativo usando como un modelo un invertebrado marino. Con Tony hemos tenido la posibilidad de colaborar desde hace un tiempo y somos muy buenos amigos. Un poquito de la historia de Tony. Tony estudió biología en la Universidad de Stanford y luego hizo su doctorado en la Escuela de Medicina de la Universidad de Washington. Luego hizo un postdoc también en Stanford, trabajando sobre todo en la estación marina que ellos tienen allá, en el modelo animal que él más tarde les va a contar. Y desde el 2010 es profesor de la Universidad de California en Santa Bárbara. Él ha publicado gran cantidad de artículos, sobre todo enfocados en el sistema de alorreconocimiento y en biología del desarrollo en su modelo animal, que muy claramente les va a explicar en un segundo. Es realmente un placer tener a Tony. Ahora voy a tratar de hablar en inglés. Tony, welcome. Thank you very much for being with us.
Of course.
You have been here physically in the past. Now
Yes.
is through Zoom, but we are very happy to have you.
Yeah.
And please have your time and show us your latest experiments, your latest ideas about the evolution of the immune system. Thank you, Tony.
Here we go. So here, I'm going to share screen. Here we go. Well, thank you, Luis, and thank you for the invitation. I'm really glad to be here. I'd rather be there, of course. And I'm going to tell you about our work today and this animal right here. And this is a tunicate that I'll explain in a second, called Batrillus schloseri. Okay, so I'll get going here. There we go. Okay. Perfect. Okay. So I know this is a broad audience, and I'm going to be talking more about immunity, but I won't go too deep in it. I really want to really pass on to you how amazing the evolution of immunity is and some of the insights we're getting from the organism that I study. So what the outline of the talk today is, what I'm going to do is I'm going to tell you about really what is the biggest evolutionary mystery, I think, and that's transplantation, the origins of our immune system, the vertebrate adaptive immune system. Then I'm going to tell you about Batrillus and its life history and what kind of insights that gives. And I'm going to be telling you mainly about two things. Most of the talk will be about a phenomenon called allorecognition, and I'm going to tell you about a unique aspect of the life history of Batrillus, and that's called stem cell parasitism, and the interplay of these and what we think this means for the evolution of the immune system. Okay, so what is allorecognition? Allorecognition is the biology of transplantation, okay? And allorecognition is the ability of an individual to discriminate between its own tissues and those of another individual from the same species, okay? How do we assess this? It's really the natural or experimental mixing of tissues, okay, like a transplanted tissue from two different genotypes. And it has two results when we do a transplantation. There's either a rejection where the interacting tissues are destroyed, or there's an acceptance where tissues fuse, forming a chimera between different genotypes. Okay. Now we think of this as often as transplantation in the clinic, about rejection of transplanted tissues and how that's controlled. But it actually goes far back in evolutionary time. And there's allorecognition in almost all the metazoa phyla that have been discovered. And what I'm showing you right here are marine sponges, okay? So if you take two pieces of a marine sponge and put them together, you'll get one of two results. Again, you'll either get a compatible, if they're compatible with each other, the two tissues will intercalate and create one chimeric tissue. If they're not compatible, they'll reject each other. And that rejection, you can see this very clearly versus here. Okay, and if you look at an incompatible reaction. The rejection reaction in this case is laying down an extracellular matrix between these two so that the tissues don't mix. Okay. Now you go up the evolutionary ladder once, and this is the cnidarians, and one that Luis works on, of course, is hydroactinia. But in corals and hydroactinia, they also do a natural allorecognition reaction. And this is a picture I took actually in Okinawa, swimming out, where two corals have come together, members of the same species, they come together, and this is an incompatible reaction, and they fire off these things called nematocysts at each other, and this is the rejection response. And of course, work from Luis on another species of cnidarian called hydroactinia also does the same thing, where two individuals come together. If they're compatible, they'll fuse their tissue together. If they're incompatible, they'll reject, and you can see this right here, this rejection response. Okay, but when most of us think about allorecognition, we think about transplantation and the vertebrates, acceptance and rejection. And this is a picture from just Janeway and Travers, this is the normal immunology textbook, and that is a skin transplant, which is put on from another individual to this individual, and you can see where this transplanted. And if it's compatible, it fuses, meaning it becomes vascularized and it integrates into the tissue. And if it's an incompatible, it rejects. And rejection is very interesting because it means that the foreign tissue, but nothing of the host, is specifically destroyed. So something is recognizing this transplanted tissue and killing it. Okay. So I think what's of interest when we think about transplantation is the model organisms used. And the first model organism used in transplantation biology was humans. And transplants have been attempted. There's even evidence, or I should say writings of transplanting from a cadaver to somebody who'd lost an arm or something, even in early Egyptian dynasties. And in 1912, a lot of research on humans was summarized in this book by George Schoen called Heteroplastic and Homoplastic Transformation. And he gave his rules of transplantation. And that was the first one, was that transplantation between different individuals usually fails. Usually there's a rejection response. But autografts invariably succeed. So an autograft is a piece of skin if you took it from the leg and put it on your arm. Those succeed. And this was important because it told you that it's not the surgery causing the rejection. There's something about the tissue itself. And this is the coolest one. And this is, the closer the blood relation between individuals, the more likely a graft will succeed. And even at that time, they had done experiments on identical twins and knew when you transplanted between identical twins, the tissue was always accepted. And of course, this was the first indication that there was a genetic basis to whether you accepted or rejected a transplant. So this is very interesting. Okay. So at that time, in the 1900s, people were just first starting to work on mice, and they were transplanting tumors, and they were transplanting skin, and they were studying it. And this is when mice were first starting to get inbred. And around World War II, research into transplantation really took off. And that was for two reasons. One was one of the biggest injuries in World War II were burn injuries. And second, antibiotics had been discovered. So people who would have usually died were now kept alive, and surgeons knew that they transplanted skin onto a burn, it would help it heal. And this pushed research into the mouse model that I showed you a second ago with skin transplantation. And George Snell and Peter Gore were looking at it genetically. And these experiments in 1951, I think this was published first, the genetic basis of allorecognition was discovered, the genetic basis. And the genetic basis was a single locus called the MHC, or major histocompatibility complex, or if people have worked on humans, HLA, human leukocyte antigen. Okay. What is the MHC? It's a single region of DNA, which is the major factor in determining graft survival. There's hundreds of different copies in natural populations, up to 1,000 of some of our MHC loci in human. The rules of transplantation, just to simplify this, the graft is accepted when both copies match. Rejection occurs if there are any differences. So for us, when we transplant tissues, the system that recognizes it recognizes difference. And I'll come back to that in a second. Okay. Now we shoot forward. Now that was the genetic basis. Nobody knew what was in the MHC. And in the 1980s, this was all discovered. And in the MHC locus, or MHC molecules are encoded. And what are MHC molecules? So I want you to think of them as a hand. The MHC is like a hand, and that hand sits on the surface of every single cell in our body. And in that hand is held a small piece of protein, a peptide, that's about 9 to 15 amino acids long. So the hand and the peptide come up to the cell surface of every single cell in our body. And the way to think about this is that that hand is displaying the protein synthesis going on in the cell. Okay. So what does this have to do with transplantation? Okay. Well, the way that our immune system works is through this hand. So under normal conditions, when the hand comes to cell surface, it's holding a piece of protein being made in that cell, okay? A self-peptide. But if you're infected with like a virus, the virus comes in, subsumes the whole machinery of the cell, starts making its own protein so that it can replicate. And now the MHC molecule, the hand, is holding something different. It's holding a peptide from the virus. And it turns out that that peptide hand, or peptide MHC, is now a different shape, a slightly different shape. And it turns out that's what our immune system sees. Your immune system knows you're infected because on the cell surface, the hand is slightly different shape than it used to be. And your immune system knows this. And that's how it decides to kill those cells. Okay, so how does this work in transplantation? If you take unrelated individuals, there's so much polymorphism out there that almost every individual has a unique MHC molecule, okay, with a unique amino acid sequence. That sequence has a different shape. So if you transplant a tissue and put it on, the immune system doesn't know it's a transplanted tissue. It just sees a different shape. And it says, that's you infected. It comes over and kills that tissue specifically if it's not a match, okay? So in some ways we think, well, this is a necessary evil about how our immune system works. That's what transplantation is. That's why we can't get over it, because we have these different shapes. We have all this polymorphism in natural populations, and that's why transplantation works, right? So it's a function of two things. One is the function of the MHC, right, as a hand, but it's also due to the number of different MHC types. If there was only 2 MHCs in the entire population, we could transplant between almost anybody, but that's not the way it is. It's very, very diverse. Okay. And that sets up the story. And that is, there's a huge evolutionary mystery about our transplantation biology and our immune system. Now, this animal doesn't need any introduction. This is a great white shark. Appears in the fossil record 425 million years ago. Great white sharks have everything, every molecule, almost everything that we have in our immune system. So they're the jawed vertebrates and the first true vertebrates, and they have a full adaptive immune system. Everything, MHC, T cells, B cells, and if you're not an immunologist, just all the cells and all the systems and everything is found in the shark. You go one step down the evolutionary ladder. These are jawless fish, okay? And they appear in the fossil record 50 million years before sharks, and they have nothing, okay? These genomes are sequenced. There's no ancestral MHC. There's no ancestral anything that's in our immune system. Nothing, okay? No ancestral genes. And I want you to just think about this for a second. For people that study immunology, it's so complex. What goes on, the interactions that go on, right? You go one step down and there's nothing. There's not even molecules that are ancestral to the ones that we use. So there's nothing we see. There's no evolutionary path we can follow. If you think about the brain or the kidney or evolution of any of those, you can follow that way back into evolutionary time. The genes that are used, Hox genes, we could go on, but not the immune system. It's insane. It is so complicated, yet it evolved from seemingly nothing. And the question is, how does the rapid evolution of these complex systems occur, right? And that really brings the long-range goals of what I think about most of the time. And that is, how does allorecognition work in non-vertebrates? What are the evolution origins of this vertebrate adaptive immune system? Are these two things linked? And is there a conserved threat that requires a genotype to discriminate between self and non-self? Is there some reason we see these allorecognition responses throughout metazoans? And the species we work on, called Bertrillus actually provides, I think, some pretty cool insight into this. Now, these are marine animals that grow in shallow waters throughout the world, mostly in marinas. They're Chordates, okay, and they're considered the basal chordate. Now, that doesn't look like a chordate, of course. The chordate characteristics come in the larval form, okay? So there's about 800 species of these. These are called ascidians, more specifically, okay? And embryogenesis results in a tadpole, okay? And that tadpole is a chordate. It has a tail, has a notochord, a dorsal hollow nerve tube, a pharynx with gill slits. And this tadpole is free swimming and swims around after it hatches, and then finds a suitable substrate, like in the case of Petrellus, it's a dock or the side of a boat, settles its head down, resorbs its tail, and then it metamorphs into an invertebrate body plan, okay? I'm gonna show you the anatomy of this invertebrate body plan. Here's the body, okay? And it's got a vasculature that exists outside of it called an extracorporeal vasculature, okay? Hold on a second, oh, I'm sorry, okay? And most of the species of these metamorph, and then they grow into these large filter feeding animals, of course, look nothing like a chordate. So there's transitioning between basically a vertebrate larvae to an invertebrate adult, okay? But there's something else with the species where we work on. So this is called Ciona intestinalis, and traditionally, ascidians have been outstanding models of development because they're like very, very fast. So you can fertilize an egg and in some species get a tadpole in 18 hours or 12 hours even. So these are excellent models to look at gene regulatory networks. You can do knockout studies in one day. So all the notochord and everything is forming very, very rapidly. So traditionally, that's what these have been used for, okay? Now, the species I work on does something very interesting, and we see this in marine animals in a lot of phyla, especially these guys that are now sessile and laying down, and that is there's a transition to what's called a colonial life history. And what that means is that Petrellus doesn't grow by getting bigger, it grows by repeated rounds of asexual reproduction. So as soon as this guy metamorphs this body, it starts budding an entire new individual. And in Petrellus, this goes on every week of its life, okay? So a few weeks after it's metamorphed, it looks like this, okay? So this has asexually budded, and then each one of those is budded. So 1 becomes 2, and 2 become 4, and so on. And eventually what you get is a colony of genetically identical individuals. Petrellus, they arrange themselves in this little structure we call a system, but it looks like a flower. Each petal is a body. This is the mouth. They share a common cloaca. They're all connected by this extracorporeal vasculature. So if you flip them over, each one of these has a heart, and they're pumping blood. And then there's a vasculature that runs throughout the entire colony, linking them all together. Okay, and you can see that here. And then on the periphery of the colony, it stops in these little protrusions called ampullae. So they're just blind-ended endings of the vasculature. Okay, and you can see these here, they're called ampullae, all right? And then in turn, the bodies and the vasculature are embedded in this sort of exoskeleton called tunic, which is carbohydrate based, and that's why they're called tunicates. Okay, so you have a solitary and a colonial life history. We're going to be talking about Petrellus today. Okay, if you look closely, and this is interesting when we get to the end of the talk. So the different colors you see are pigmented cells in the blood. So about 8 blood cell types. So I showed you blue. This is an orange colony. So here are the bodies, and these little orange tips, these are actually the ampullae that you see at this magnification. If you look closer at the anatomy, they're very complicated. This is the muscle shown by Floyden. They have a central and a peripheral nervous system. We're just seeing this without the tubulin. There's a little brain right here. And again, here's the vasculature. We look at a cut through of the anatomy. They're very complicated. It's like the GI tract, incurrent and excurrent siphon, heart, primitive endocrine glands. They're very complex, okay? And they also have a germline. We'll be talking about that. So each one makes its own sperm and eggs, each individual, okay? And I should tell you one other thing that's really cool about Petrellus, very unique. And you'll know this if you're familiar with Luis's work, because hydroactinia is like this too. And that is each one of these bodies are linked together normally, but they're not dependent on each other. So you have a cool experimental trick you can do. You can cut a piece out, transfer it to another substrate, and it'll continue to grow. And that's called subcloning. And we'll use that later in this talk. Okay. So this is what it looks like. Tadpole becomes a single individual. It starts to bud, and a few months later, this is what you have. Okay. So this gives you an idea of the sizes. Each body is a few millimeters. So this is 1,000 bodies. Each one of these is about 12 bodies that are independent bodies, but this is all one genotype. And again, you can cut pieces off and transfer them, and we grow them on glass slides in lab, and you can transfer them to a new glass slide, and they'll continue to grow. So we can have hundreds of naive pieces of the exact same genotype. We can study a genotype as it ages. This is a very powerful aspect of the life history that we can utilize. But what I want you to take home from this, so they're growing all the time. So development's always occurring. Every week, each one of these bodies is giving rise to 1 to 4 new bodies. And an individual expands over the substrate. But importantly, development is always occurring, unlike most other organisms. So every week, Bacillus is regenerating a new heart and a new brain and a new stomach and so on. Development's always occurring. Okay. All right. So what does this have to do with allorecognition? So imagine this little tadpole swims around, settles down, and starts to grow and expand over the substrate asexually. And as it does, it's growing over the substrate. It's going to bump into other members of the same species. And this is a picture, and this is really beautiful because you see, for you evolutionary people out there, there's all kinds of polymorphism at the colors of this. So these are all just differential pigment cells and homing to different places, but these are all Bacillus schloseri. And this is a settlement plate that we put out on the East Coast of the US in a place called Woods Hole. And these are growing into each other, and these are all Bacillus schloseri, and they're going to touch each other. And as they grow out, the first thing to touch are the ampullae, okay? And that's shown right here. And I'll give you a close-up. And when the ampullae touch, this initiates a natural allorecognition response in Bacillus. So the ampullae touch, this initiates an inflammatory reaction, and you see these cells come into the ampullae that are in contact, and then one of two things will happen. If they're compatible, the two blood vessels will fuse, forming a chimera, a parabiotic chimera, basically a hematopoietic chimera. If they're not compatible, they reject. And in the rejection response, blood cells pour out into the region in between the ampullae, and these ampullae become leaky, basically. These are called morula cells, and inside them, they have the precursors for a biochemical pathway called the prophenol oxidase pathway, which turns tyrosine into melanin. And these melanin scars, we call them, or points of rejection, are laid down. Okay, and this prevents any blood transfer. So you fuse and you share blood, or you reject and you don't share blood because of the scarring. Okay, so these are huge. Okay, these are a couple millimeters in length, and you can just watch this in the dissecting microscope. And from touching to fusing or rejecting takes about 24 hours, or 24 to 48 hours. Okay, so it's a very nice thing. It's a very nice system. But why is this even interesting in terms of vertebrate allorecognition? Well, we already have this phylogenetic relationship that these are the basal chordates, right? But it was the genetics of allorecognition of Bacillus that made it really interesting. Okay, so whether an animal fuses or rejects is also due to one single locus in the genome called the FUHC, for fusion histocompatibility. Okay, it's highly polymorphic. Okay, there's about 100 alleles in the Monterey Marina, and we're in Santa Barbara. This is where I did the work originally in Santa Barbara. There's several hundred. We think there's about 1,000 alleles of this locus worldwide. Okay, but so this is very familiar from the MHC part of this talk. But the rules of FUHC-based allorecognition are different. Colonies fuse if they share one or both alleles, and rejection occurs if there are no alleles in common. This is in contrast to the MHC-based allorecognition in us, where you only accept if you share both alleles and you reject if you don't share both, right? So in our model, in humans, you see the system sees a difference. But in Bacillus, it sees self, okay? And I'll come back to that in a second. And the question, of course, is given this phylogenetic relationship and given this genetic similarity, is the FUHC the ancestral MHC? Okay. So when I started postdocing, that was the question I took on. And I came in a lab of a guy named Irv Weissman, who was very interested in this and had started doing basically what was done with the mice, almost 100 years ago. And that is, he started inbreeding. He had lines of Bacillus grown in the lab. They undergo their whole life history in the lab. And here they are growing on these glass slides again. So we could cross them and raise them in the lab. We had strains with known fusibility types, okay? And we can also study wild type genotypes. So I just want to show you this here. So in the marina, they're often growing like this is a mussel shell, and we can bring them into the lab and transfer them onto glass and they'll grow, okay? But because we could do these two things, we could carry out forward genetics. So I just took a classic forward genetic approach to positionally clone and assess what was in the FUHC locus. And this is what's shown here. So these are genetic markers. This was a long time ago. Now I can do this in a day, but it took me many years. But after the crosses, I should say. But these are clones that we rebuilt the FUHC region of the chromosome. Okay, and the business end was over here in this region that was about 550 kb in length. And there we found 6 proteins. Okay, that look like they might be involved in the allorecognition response. So what were we looking for were proteins that were highly polymorphic. And these ones with the stars are highly polymorphic. So these are the molecules right here. Okay, I'm going to tell you about these two and these two right now. And polymorphisms in these molecules right here are segregated with fusibility. Okay, so the 2 most polymorphic proteins in this region, okay, were encoded right next to each other, and they're called FUHC-seq and FUHC-TM. Okay, so again, when we looked in the locus, I guess I didn't say that, although I had it here, FUHC is not the ancestral image locus. It's something completely unique. Okay, and these molecules that we found encoded in this locus are also completely unique and are not found in other organisms. Okay, so there was 2 right encoded next to each other, only 247 base pairs in between, and that was FUHC-seq, which has an N-terminal signal sequence and 2 EGF domains, and there's FUHC-TM, which had an N-terminal signal sequence, 3 immunoglobulin domains. And the reason I'm pointing that out is most of the immune genes in the vertebrate immune system are immunoglobulin based, and this is just a way that the protein folds. Anyway, there's 3 immunoglobulin domains, a transmembrane domain, and a short carboxy tail inside the cell. So these were the 2 most polymorphic. And when we looked at polymorphism in these proteins, so this is called this is how we assess polymorphism. Okay, so the FUHC is about 450 amino acids. The TM is about 250 amino acids. And what this is showing you, this graph right here, is how the polymorphism is distributed along the protein. So each one of these is a residue, and each dot resembles how many different alleles are there at this amino acid, how many different substitutions. So the higher it is, the more substitutions out there. So you can look at this, and what you see is that if you look at a bunch of alleles, the polymorphism spread throughout the protein pretty much looks like randomly. If you look at any 2 FUHC-seq alleles, they differ by about 20 amino acids that are all over the protein. Okay? Same thing for TM. It's not nearly as polymorphic, but we kind of see the same thing. And if we look at these highly polymorphic residues, a lot of them under natural selection. So we can see signatures of natural selection on them, right? So there's selection for polymorphism, which is, we'll come back to again. Now, if you compare this to proteins in our immune system, like the antibodies or the T cell receptor or the MHC, they often have regions of very, very high polymorphism, and that's shown right here. So this is completely different. Why am I showing you this data? Because we want to understand, how does this system discriminate between 2 alleles? How does it know what's an allele, what this allele is and what that allele is? How does it know this is a self allele and this is a non-self allele? How is that accomplished from a biochemical standpoint, right? From a structural standpoint. And this is what really excites me. So how is it discriminated? How does this system work? How do they come and go, oh, you match with me or you don't match with me? How does it know? Okay. And the question is, how do you discriminate between these alleles? Okay. So these are 3 hypotheses. Well, it's one hypothesis, really. One is that it's homotypic. Okay. And that is that allele A on this MPUA only binds allele A on this MPUA, and that's how they tell self. And allele A doesn't bind to allele B or C or D or E. Okay. So a homotypic binding, and B binds to B and C binds to C. I find this not to be even possible, because that means that during evolution, you would make some amino acid changes, and then they'd only bind themselves and nobody else. And I can imagine that for 10 alleles, but if there's 1,000 out there, how about the 800th allele, right? Which means it doesn't bind. Every time there's a new allele, it has to be even better at binding itself because there's even more of them. So that doesn't make sense. Okay. So in Bacillus, there's no recombination. Okay. In our immune system, if you're aware of this, we take loci and we diversify them, and we tailor alleles, and we tailor receptors to bind to the MHC in that individual. We don't have that in the system. So we're not making a receptor specific for each one. Okay. And the single naturals, meaning that we fuse, when two MPLA come together, they're looking for one self allele in that other MPLA. And if they see that, they'll fuse, and if they don't, they reject. These rules are telling you, you're not recognizing the non-self one, you're looking for the self one, okay? So this is called missing self-recognition, okay? So the idea is, when the MPLA come together, they're looking for a self allele on the other one. So if they have two, A and B, they're looking for A or B. They don't care. Either one of them, if they find it, they're compatible with, okay? A single allele match, then, if we say that, blocks the default rejection reaction. I'll tell you what that means in a second. And we didn't think of this, of course, because this was already seen. There's part of our immune system, not the adaptive part, but another part, the innate part, that does this missing self-recognition, okay? And there's a set of cells in our immune system called natural killer cells, and they have two types of receptors that recognize the MHC. There's another completely unique system called NKRP1B and O cells. So O cell is the ligand, like the FUHC is the ligand, and these are the receptors. And there's another one called SIRP alpha, CD47. And this CD47 is a cool story because this is the way old red blood cells are killed when they go through the spleen and the sinusoidal macrophages. So if a red blood cell is healthy, it has high CD47. When it goes through the spleen, it's fine. If it has no CD47, those cells get eaten, and CD47 goes down as they get older. So if you're familiar with this, that's missing self too. Okay. So how does this work? And we actually got a clue because of genes encoded in the locus. And the first gene I found that was actually polymorphic was a gene I called Fester. Okay, and Fester, again, had no relation to anything in the databases. It has a signal sequence, a domain called the sushi domain, okay, and 3 transmembrane domains. This is what it looks like, a structure. So it's N-terminus, SCR domain, 3 transmembrane domains, and a carboxy tail. Okay, so this is the first gene we found. There's no signaling domains or anything on that tail of Fester, okay? But Fester was polymorphic. In fact, there were 3 levels of polymorphism. Genetically, there are about 80 alleles, but this was interesting, and we knew this in the cross. Polymorphisms didn't contribute to outcome, meaning you could share a Fester allele and reject or not share one in fuse. So it was polymorphic, which was interesting, but it wasn't determining whether they fuse or reject. Genomically, there's haplotypes of this, meaning there's different chromosomes out there. Some have 2 Fester locus, some have 1, some have 3, and I'll talk about that more in a few seconds. But what was really interesting too is when we were cloning this from different individuals, we found there was another level of diversity, and that occurred through alternative splicing. And what happened was that Fester was encoded in 11 amino acids. 6 of these can be alternatively spliced, and we've seen all variants, all combinations of those 6. So you can make 64 different proteins from this single locus. And what we found that was really interesting was that each individual made a unique repertoire of splice forms. So every individual made a full-length form, and then a couple different splice forms, 4 to 24 different ones that were unique to that individual. And I'm not going to show you this data, but they were maintained over time. So somehow they're alternatively splicing this gene, creating a repertoire of splice variants that's different between individuals, but stable through that individual's life. So this was really cool. So what did we think? This is what we were thinking, that somehow these Fester splice variants, which I've got outlined here, somehow together would bind to FUHC, and they were somehow figuring out how to splice them so that they fit the FUHC that was in that body. Okay? And this is a process called education, right? But what they're doing is setting up an individual recognition system. They have a ligand and they have a receptor, and we think this is how this was going on, basically because we saw this difference between individuals that was maintained over time. Okay. So our guess was, if you think about signaling or binding, was that these different splice variants together signaled, and the avidity was somehow determined by the cell, whether that was self or non-self. Okay. So we made a monoclonal antibody to Fester, and I just want to show you over here. So when we made the monoclonal antibody, this seen in red, this is an ampullae counterstained with DAPI. So these are blood cells. This is out in the tunic. So Fester was expressed in the ampullae, at the tips of the ampullae, as well as a subset of blood cells. And the first experiment we did was this old school monoclonal antibody binding experiment. And that is, we injected the monoclonal antibody into animals as they were coming together to fuse or reject, and we got this crazy result. And that was that If two animals were fusing and we added the antibody, it had no effect. But if two animals were rejecting and they both had the allele we made the antibody to, we could turn a rejection into a fusion. And this is really cool. So you can see these rejection going on right here, and right here, fusion. So we could override a rejection response by having the antibody, which would turn it into a fusion. So our idea was, oh, that's great. This is right. And the antibody is somehow binding to these such that they think they're binding to self. And that's how we turned it into a fusion. Okay. So this is called an inhibitory receptor. We're mimicking the binding. Okay. And we're turning a rejection into a fusion with this antibody. So we're overriding the rejection in progress to turn it into a fusion. Okay, so I wish we had stopped there, but we didn't. And during that time, we had the monoclonal antibody, so we were cutting these blood vessels off and doing FACS and stuff like that. And we found that they would regenerate in about 48 hours. So there's a lot of angiogenesis that goes on. So they immediately clot and they regrow. And then at the same time, we had been experimenting with doing transient knockdowns using siRNA. Okay, we found if we injected siRNAs, they would be taken up. And you could basically, and this is Fester here at the qPCR, in 3 days, completely knock out the expression of the mRNAs. Okay, so we could destroy the mRNAs. So this means we can combine these two, because this is an adult animal and we don't have transgenics. But we could combine these two assays. So we could cut them off while they were in siRNA and force them to regenerate. And when we did, we could force them to regenerate without Fester on it. So this is a control siRNA and a Fester RNA. So these ampullae have no Fester. And when we did that, and then we paired up two individuals with no Fester, we expected to see rejection responses because we thought this is how fusion was mediated through this, but that's not what we saw. We saw something completely different. And that was, we saw no reaction phenotype. So these guys came together and touched, right, and did nothing. They just sat there as if they had hit a rock or if they had hit, some other animal. They just sat there because the out recognition response goes on only between two individuals. I'll talk about that in a second. So we got a completely different result. We were very confused, but we thought, oh, Fester must play a role in both rejection and fusion. So Fester's polymorphic, it's somatically diversified. We suggest that it binds to the FUHC. That's one piece of data, but another piece of data says it also plays a role in the rejection response. And that brought up the question, what's the relationship between fusion and rejection? And again, we got lucky because we found something very weird. And that was about 60 kb away from Fester was another gene we found. And the back end of it that encoded the transmembrane domain is almost 100% identical to Fester, even at the DNA sequence. But the front end of the gene completely divergent. We would never see this, but it encodes the same exact topology. The other great thing and very interesting thing about this is Uncle Fester was not polymorphic. So we cloned it from Europe and from Japan and from the US, and it's this almost the exact same amino acid sequence down, but it's the same exact structure. So because of this half relationship, I called it Uncle Fester, TO Fester. Okay. We did the same set of kinds of experiments. We knocked it down. And then this was in colonies that were going to reject. So when colonies were coming together, they were going to reject, and we knocked Uncle Fester out. There was no rejection response. Okay. Just like we saw with Fester. But the surprise came here. When we knocked it out in colonies that were compatible, it had no effect. Okay. So Uncle Fester was involved in rejection, but not fusion, right? In contrast to Fester, which was involved in both. Okay. And this was really cool because it told us fusion and rejection are not mutually exclusive. They don't come together, we're going to fuse or reject. There's two different pathways controlling fusion and rejection. One goes through Uncle Fester, and one goes through Fester. Okay. And then this was work by a grad student in the lab, Connie Kittrick, and she did a beautiful experiment. So she made a monoclonal antibody, also found Uncle Fester was expressed on the ampullae. She put the monoclonal antibody onto magnetic beads, dropped it down into the water and there's a Bacillus colony, and then used a magnet to pull the beads over and touch the ampullae. And when she did, with this one of the Uncle Fester antibodies, she could induce a rejection to a colony just sitting there. She followed it up, and here's a control. So you can see these guys undergoing this inflammation after they've been touched. This was with a control antibody. There was no effect. But she did the opposite experiment of what went on with that had been done with Fester. And that is, if two colonies were coming together and they were going to fuse, she laid, in this case, the antibody over the interface of the ampullae, and she could turn a fusion into a rejection. Okay? So uncle Fester is non-polymorphic family. It's coded near Fester. It initiates a rejection, but has no effect on fusion. So these two pathways are different, right? There's two different pathways controlling, one's controlling fusion and one's controlling rejection. And somehow these two pathways are being integrated, okay? So we can change the severity of the rejection. We can manipulate this by these monoclonal antibodies, and it suggests that signals from two independent pathways are integrated. Okay, what's going on? Why am I telling you about all this? Okay, so let's go back to the beginning. There's no conservation at all of anything in our immune system in lower organisms. Okay, there's candidate molecules in sponges and as you know, Luis, in hydroactinia, there are candidate molecules involved in Al recognition in those animals. They have no relation to anything else. Nothing in the vertebrates, nothing in other organisms. In FUHC, we find it in other petrilids, but no other tunicates or any other invertebrates. Okay, so each system is unique. What does this mean? How is this even possible? Okay. Now they're unique, but Luis and I were just talking before the talk. But there's two familiar characteristics that we see both in Bacillus and hydroactinia that are very familiar. Okay, one is genetic, that there's linked receptor families which are polymorphic and polyjack, meaning there's haplotypes with different numbers of genes in them of different receptors. And functional, the outcomes are determined by integrating two pathways, one which turns the pathway on and one which turns the pathway off. Okay, so genetically, if we look in the vertebrates, there's actually receptors that are involved in recognizing the MHC. I told you about these are natural killer cells. And when we look at how they're organized in the genome, they are in these big, huge clusters linked right next to each other, okay? And there's a lot of different ones. I'm showing you these pictures from Jeff Yoder's lab, who's looking at these genes called NITRs. Okay, these are all immune receptors, okay, and they're in this organization. That's conserved. That's what we see in controls. Okay, and functionally, what resembles what we're seeing is that it turns out the immune system works in the same exact way, no matter what kind of immune reaction we're looking at. There's activating and inhibitory inputs, okay? And these activating and inhibitory inputs, very interestingly, are linked and signal through the exact same signaling domains. What activates an immune reaction goes through what's called an ITAM domain, which is on cells, on the receptors. And what stops it is called an ITAM domain, which is on other kinds of receptors. So there's something that turns on a reaction that signals through an ITAM domain, okay? And this is a kinase cascade. And there's something inhibitory that signals through a phosphatase, which shuts this off. So these two signals are integrated, okay? And this is what I'm showing you for NK cells right here. So the same thing we're seeing. Uncle Fester turns on the reaction, Fester turns off the reaction, okay? Now, if you look at these, what you see is in this linkage in the vertebrates is activating and inhibitory molecules put right next to each other. Okay, we see this all the time in a bunch of different organisms, all the vertebrates. And really, Fester and Uncle Fester are kind of a scaled down version of that. So Fester and Uncle Fester are linked to the FUHC. There's polymorphism and polygenism here. So there's different haplotypes. Okay. And actually, Henry Valbuena, who came from Luis's lab and is doing his PhD in my lab, has found this is like even way more complex. There may be 40 Fester family loci. And in one animal, he found 10 different ones. So what we're seeing is something very similar to what we see in mammalian. So what does that bring me to? And this is what I wanted to tell you about today. So there's two hypotheses when we look at this data. One is that high polymorphic allorecognitions can just evolve convergently, okay? And I just want you to think about how kind of exquisite this is, how you can discriminate between these alleles. And we know that the Petrillus can discriminate between two FUHC-seq proteins that only differ by 4 amino acids. So it can discriminate between a self and one that's only 4 amino acids. How does that occur? So either that's easier than I think, and I'm totally mistaken, or we've been looking in the wrong place for conservation. Because what we did find as genomes started getting sequenced not long ago is that immune signaling pathways, these ITAM and ITAM, I told you, are conserved. And they're conserved all the way down to sponges, which have orthologs of the genes used in our immune system, okay, that are in our adaptive immune system. So all of these are found in Bacillus, and these are, you don't need to know these. These are just signal transduction molecules found in these different organisms that are key parts of how our immune system functions in humans, right? So our hypothesis is that this is what's conserved. Okay, it's the intracellular wiring, right? The intracellular wiring is conserved, but the intracellular wiring is conserved, and the receptors, they can evolve. And there's a great example. And again, I didn't come up with this. There's a great example that came out while I was doing my postdoc, and that is in mammalian natural killer cells. And it's a crazy story, whereas in mice, the cells in natural killer cells, the receptors in natural killer cells are C-type lectins. In humans, they're immunoglobulins. They're not related at all. But the tails are exactly the same, and they have these ITAM and ITIM domains, okay? So what I'm telling you is there's a mix and match of ectodomains to the same intracellular wiring. And what I think is conserved is that integration. If the wiring's conserved, right, then you can just let the receptors evolve. And this is very important for an immune molecule, right? If it's got to evolve, if it's recognizing things to kill or not kill or self and non-self, if that's linked down to a common wiring program, then this allows it to go. So it's not the receptors. We've been looking for homology in the receptors and the ligands. And what I'm saying is that doesn't matter. What matters is this, okay? It's the intracellular wiring. And why is that cool? Because this has significant clinical implications, okay? Because we don't know when a T cell binds a target or a natural killer cell binds a target or any kind of immune reaction goes on, we have no idea what's counted. How's the threshold set? How does the cell know that it should respond or not respond? How does it set that threshold? And if you're a developmental biologist out there, you're like, oh yeah, whatever, man, you know, Wnt and Notch turns this neuroblast into this kind of nerve cell, right? But the integration here and the thresholds are very much more complex, and they have serious clinical implications. And that is, how do you maintain that? Because if you have a threshold about when you're going to react, why do people get multiple sclerosis when they're 30? The threshold's changing. What happens? Like one of the biggest things in cancer now is checkpoint therapy. Checkpoint therapy are molecules that bind this part of the signaling when T cells become exhausted, okay? And what I've just showed you is why Bacillus is a great model to look at this. And we have not linked signaling to the ITAM or ITIM domains yet, but we have these in the ampulla, okay? But there's two inputs in Bacillus. In mammals, there's hundreds of these receptors. We don't know what they bind. They're expressed on all the cells. And basically what I'm saying is there's a very complex interplay, all kinds of receptors for one of our immune cells to respond that has clinical implications, because if we could just control that, we could control somebody who was autoimmune, we could restore a T cell that's exhausted, that's fighting cancer, right? Because that's all about this integration and how those thresholds are set. Okay, so I know I'm probably, I'm gonna be very quick here. Okay, so that's the idea. So the idea is long ago, cells learned how to count what was going on the outside of the cell. And they had wiring and they knew how to do this. And this is what controls allorecognition. So if we've been looking for an ancestral MHC molecule, the ligand and the receptors, but that doesn't matter because they can just evolve, and it's what's inside the cell that matters. Okay. So that's that part of the immune system. Now I want to tell you about why is there allorecognition and everything? Why are all these metazoans? Why can you transplant and get these rejections, or why do you accept, right? And more specifically for us is, why is there allorecognition Bacillus? And I'm going to finish up quickly here, but I kind of started this showing you this already. So again, tadpole turns into this, and then it starts to develop, starts to asexually reproduce. Every week in Bacillus, they're reproducing whole new bodies, heart, brain, and the germline. Okay. Now each new body makes its own gametes. So this is a picture of a zooid from the bottom. You can see the vasculature. These are testes, okay, growing in this animal. Okay, so each one makes new bodies. Okay, and what this implies, right, is that there's some cells, and if you're a germline person out there, a developmental biologist, Bacillus has germline stem cells, okay, that are very, very long-lived. and give rise and seed new zooids as they develop and give rise to the new germline. So how do we know that? We know that because of experiments in recognition, and this is a great story. And this was discovered by Armando Sabatin, who's at the University of Padova in Italy, published this in 1982. This is beautiful. So we know about germline development because of what happens after two compatible colonies fuse. I've got this in cartoon form. So here's the zooids, and these little things are the testes. These little circles I have represented as the testes. Okay. So what happened after parabiosis of two different individuals? If you looked in the zooids that developed after they fused, what you would often see is chimerism, and that is the blue colony would be making testes from the orange colony, and vice versa. The orange colony would be making testes from the blue colony. And this suggested there was a mobile progenitor for the germline, and that it could cross over between the fused vessels and go into the other individual and contribute to development in that other individual. This was chimerism. But we also saw something, he also saw something remarkable, and that is in some pairs of genotypes, one would win. Okay? And all of the germline, in this case in the blue colony, would come from the orange genotype. And this would be true for the rest of the life of that colony. So basically what's going on is the stem cells for the germline from this individual are parasitizing the other individual. And even if we cut them apart and separated them, they'd be true for the life of this colony. So it would continue to somatically grow, would be making the germline. So basically it's dead in an evolutionary sense because its germline has been taken over. Okay. So germline stem cells can naturally migrate between fused individuals. And what we found, this was followed up by a postdoc in Irv's lab that was finishing when I started, and that's the winner and loser genotypes are found in nature, and parasitic abilities arrange itself in a transitive hierarchy. So I just want to walk you through this experiment very quickly. So in this experiment, we have three genotypes, F, B, and G. Okay. And again, we can cut these pieces, we can cut the genotypes apart and have natural, naive pieces of the exact same individual. So if we fuse up, if F and B are compatible, we fuse them 1 time, 2 times, 3 times independently, and we look at the genotype of the sperm following the fusion. If F beats B 1 time, it beats it every time. If G beats B 1 time, it beats it every time, and F, G here. But what we found from these experiments, where we really were randomly picking animals out of the mariculture, is that the ability to parasitize actually arranges itself in a transitive hierarchy, meaning if F beat G and G beat B, we knew F would always beat B. So you could predict the outcome based on previous matches between these at the germline. Okay? So this was very interesting. We see this a lot. Okay. So what did we do? We've taken, now we can repeat this, and I just want to show you this one slide. So we take the animal and make single cell suspensions and then inject it. We can recapitulate this and see the germline come from the donor into the recipient. And in these experiments, is what we found was that the ability to parasitize was autonomous to the stem cells. Meaning, if you took cells from a winner and transplanted them into a loser, they would expand and differentiate and give rise to testes. But if you took them from a loser and transplanted, so these are the loser cells, transplanted them into a winner, that would not happen. Okay, so transplantation, recapitulated parabiosis, the ability to parasitize is a property of the stem cell itself. Okay. So we have an allorecognition system, which is basically preventing or severely restricting the movement of stem cells among animals in a population. Okay. So the transplantation is there to block movement of stem cells, we think. And those stem cells are deadly because they kill you evolutionarily, they can take you over. Okay. So is this a quirk of Bacillus, or is this evolutionary important? So I think it's important. We know that there are transmissible cancers. And I should say that in the early days of MHC biology, well before we knew about what it was at a molecular level, one of the ideas around was, well, heck, if we didn't have this transplantation biology, if we didn't have all this diversity, cancers could be transmissible. And they are. There are examples in the mammals. And this is the Tasmanian devils, which have a facial tumor that when they fight, it's transplanted. And if you were a wily, mean cancer and you really think about how would I move around a population, you'd say, well, I'd be sexually transmitted. And there is actually a sexually transmitted cancer in dogs. And they think the cancer itself is about 40,000 years old and it moves around. So this is found in wolves. So if we didn't have transplantation biology, it's possible that cancers would be transmissible. So maybe that's the evolutionary force that drives both the appearance of this, but more importantly, the polymorphism, that the molecules are polymorphic, meaning each individual is slightly different. It has has its own, we all have our own self, and it's pretty much unique. I mean, the chances of any of us matching MHCs with another person is more than 1 in 20,000. So each of us is pretty much unique, okay? So this system, like in Bacillus, is maintaining integrity. Now, if we go back, there's transmissible cancers apparently. So this is a paper from Nature, even down into bivalves. Okay, so let me just summarize my talk here. Allorecognition exists, and it exists throughout evolution to protect an individual from a parasitic cell lineage, because that's what a cancer is. It's a parasitic cell. I think this has been a selective pressure throughout time, throughout evolution, from the very origins of the Metazoa. There's great stories about that. And then why are they polymorphic? They're polymorphic because there's an evolutionary battle between the parasite and the molecules used to detect that parasite, right? Parasite says, this is a red queen, right? Parasite changes, receptors need to change. Parasite changes, receptors need to change. Pretty soon, there's all kinds of polymorphism in the system, and that's what's driving this polymorphism. And that's why we see not just allorecognition, but highly polymorphic allorecognition throughout the metazoa. So we think maybe this is the pressure that's pushed this, right? And then once you've done that, then you use the system for immunity or, detecting other people you might be attracted to, and so on and so forth. Okay, I'm going to finish there, and thank you. And I'm sorry, I know I went over a little bit. So this is a picture of UC Santa Barbara. Our lab is right here. So this work was really done, a lot of this work was done by Spencer Nyholm, who was a postdoc in the lab when I was at Stanford, and Tony McKittrick. Henry, who's from Columbia, is doing a spectacular job here and really digging into the Fester-Uncle-Fester evolution. Adam Langebacher and Stefano Tiozzi also contributed to this, and Greg Stoney is our lab manager who maintains our animals. Stanford, I started in our Weissman. This is our funding, and some other of our collaborators who have really helped with this project. And I'll finish there, and I'm happy to take any questions.
Thank you, Tony.
Sure.
Great talk. Thank you very much. It was very interesting.
Not too long.
Para la audiencia.
Only 8 minutes. I mean, that might be a record for me.
That's a record, certainly. Para la audiencia pueden enviar sus preguntas a través del chat de YouTube. Pueden también enviarnos un correo a la dirección bioinfoigen, radialpisobogotá.unal.edu.co, o hay un Google Docs, Google Forms que pueden llenar para hacer sus preguntas. Mientras se van animando a hacer las preguntas, quiero empezar. I'd like to ask you a couple of questions, Tony. The first one is regarding of solitary tunicates versus colonial tunicates.
Yeah.
Have you seen the same genes that control recognition in colonial tunicates in solitary tunicates?
So this is Henry. Yes, Henry is doing a full-on looking through all the genomes as they're going. We're actually doing some sequencing too. So Henry has found this beautiful, really fascinating distribution of these among species. So he has found in a solitary an FUHC-seq that's not polymorphic. We have no idea what it does. He just found this. And this is very interesting because this may, from an evolutionary standpoint, this was maybe a fantastic and huge finding, because we don't see any animals with non-polymorphic, well, we don't see any species that really have, well, maybe that's not true either. But polymorphism is a key part of the MHC, I should say. Some species have different levels of polymorphism, and it's different in different species. But in this case, we don't see any polymorphism. He's also looking at the evolution of festers and uncle festers, and they only are in the colonials. But other molecules in that locus are found in the solitary. So, I don't think he has the time. Unfortunately, I don't think the species he found, the sequence was done, but I don't think there's experimental tools to look at what it does, but we can at least try to see where it's expressed. and see if it had a role prior. Because, you think about this, Luis, and all the evolution that goes on. I mean, they're just they're whatever the system is going on. When we think about evolution, let me see, there's all kinds of ideas about where it came from and what it did originally and how it turned into what it is. And at some point, the idea was non-polymorphic. And some species that's, I talk about evolution, but we only look at extant genomes, right? I mean, a lot of things went extinct in those 50 million years, and they must have had precursors to this, but we just don't have any clue in anything that's alive anymore in those genomes. So, yeah.
And another question related to that is, what happened in the tadpoles with the expression of those genes that are involved in allorecognition?
They are. So, and I didn't talk about this, but when I was looking at molecules, so basically we're positionally cloning this locus. We have the genetics, and we're looking at what then were large insert genomic clones as we rebuilt the chromosome. And as I looked at, I was looking for something polymorphic, but another characteristic that I thought had to be true was that they were expressed in the tadpole. And the reason is that, hold on, as soon as the tadpole metamorphs into this body plan, it's competent to undergo allorecognition, okay? And it's highly specific. So the whole system has developed by the time they metamorph. So when I was looking at genes, they had, to me, they had to be expressed in the tadpole, and they were. So Fester, FUHE, all of those are expressed in the tadpole.
Okay, there's a question from Adelaida Turriago. She asked whether there has been any reports of any type of cancer transmission between humans.
No, not that I know of. Okay, so no, not that I'm aware of. But I will say that there is one time there's a natural transplantation in humans, and that's during pregnancy. And there are self-renewing cells from the fetus that transfer into the mother. And there are cases where the mother will get an autoimmune disease that correlates to the father's MHC. So, yeah, I think this is a real thing in humans too. Although that live birth and everything evolved long after the immune system did, right? But yeah, so I can't say there's natural transplantation of cancers, but there's certainly natural transplantation in humans. And that occurs during every single pregnancy, which is obviously something under very strong selection.
Sure.
Right, and the other thing, I think, if you don't mind me adding, please remember that the other kind of really cool thing when I talk to you about thresholds and integrating data and moving thresholds is the fetus is a graft that should be rejected. And during pregnancy, if you transplant a piece of skin from the father into the mother, she'll reject it. Yet the fetus is protected, and that's tolerance, right? You're telling that somehow the body's telling the immune system, do not kill this fetus that's growing inside, right? And this is really fascinating, right? Yet cells are moving and not getting killed. It's really interesting. Anyway.
Yeah, thank you, Tony. There's another question from Nicolás Alejandro. He asks whether allorecognition is exclusive of animals or is a phenomenon that is widespread in multicellular organisms.
There is, yeah, so the other place you see this is in plants and inbreeding. So many plants are hermaphrodites, and they have a block to self-fertilization. And I think it's in mustards, and it's called the SI system. So they can recognize the difference between self and non-self pollen. And they'll kill self pollen, but not non-self. Yeah, so you see this in other, and I guess it's kind of the take-home message of that is you see allorecognition in other phenomena besides immunity, right? And that would be preventing inbreeding, right? So that's a multicellular thing anyway. But But point is, yeah, there's other times you want to discriminate between self and non-self besides just an immune response. And when one of those would be to prevent inbreeding.
We have another question from Nicolas Romero. He asks, you show some examples of somatic diversification through alternative splicing. And he asks whether there's any evidence of somatic hypermutation in those genes involved in allorecognition.
No, we've never seen anything like that. There is evidence of that in another invertebrate, in these snails, in these genes called FREP genes that I know Luis is very familiar with because he worked with some people with that. So there is evidence of that, but nothing, we've never seen anything like that in Petrellus. And we've looked at so many individuals and so many genes, and done the genetics, and we don't see anything changing in terms of individual specific, like somatic hypermutation or recombination or anything like that.
Okay. I have another question, and I was really interested in the model that you are proposing about the Uncle Fester as a mediator with these different splicing forms, as a mediator of avidity in the
right.
FUHC
Right.
union with this ligand receptor. How does it work? How does it change its avidity?
Oh yeah, no, I know. Well, okay, so this goes to your other question too, Luis, because that's why I had to see genes expressed in the tadpole. So this is a process called education. And that is that our immune systems are tailored to our MHCs in humans. And that goes on through a process called education. And basically what happens in us is that the B cells, let's talk about the T. The T cells in our body, which recognize the MHC, each cell makes its own unique T cell receptor. And this is just the receptor that binds the MHC. And each cell recombines and randomly makes a T cell receptor. And those cells go into the thymus, and thymus is like school. So in the thymus, you get educated. And basically, you need to bind to your own MHC. And if you don't, you die. Okay? And that's called education. This goes on in natural killer cells too, in a slightly different way, but the same thing. And natural killer cells, we think is more like Petrellus. Natural killer cells also bind MHC, and they have a bunch of receptors in one of these clusters I told you about. And what they do is when they're developing, they send one up stochastically, they send another one up, and they wait till they find the one that binds to the MHC. So I think in the tadpole, somehow there's a time where there's plasticity. So they're just making a bunch of splice variants, waiting till they make the right recombination, the right repertoire to bind to the self-FUHC, and somehow that's locked down. Now that's complete speculation. But the way to look at this is we're going to start looking soon at epigenetic marks along the Fester locus from genotype to genotype to see if we see any evidence of. So basically, I think it's plastic. Then they get the right signal, then it locks. Same thing happens in T cells. Same things happens in natural killer cells, where so part of their development is like, I got to bind to my ligand, and if I don't bind to my ligand, then I die. Right. And that's education. So we think the same equivalent thing goes on. So I think that's conserved too. And that's somehow in the signaling pathway, I think.
Okay. Yeah. There's another related phenomena in NK cells, which is priming.
It's
sort of a
same thing. Yeah.
Some people call it like memory in innate immunity.
Yes.
So my question is there any evidence for memory in the allorecognition response, like a second instance contact with a colony is going to be faster or stronger than the first one?
Never seen evidence of that, ever. It's always pretty much the same. And that I didn't do those experiments, but they were done by a lot of people. And there was never any real evidence of memory. And I think, in some, I know that there's like epigenetic memory and the innate immune system of some organs that's been discovered. And I wouldn't be surprised if there was, I just don't think we have, I don't think, it's nothing we've ever seen any evidence of really at all.
Okay. And finally, Tony, and thank you again for your time and disposition. What's with these secreted forms of the allorecognition molecules?
Yeah.
How do they work? I mean, they are secreted and they are contact.
No, I think it's assembling. I think it's like. I think it just assembles with the trans. I think the two transmembrane and FEHC assemble into a heterodimer. That's what I think, right? Because if you secreted them, that makes no sense, right?
Sure.
I've always wondered what that even meant. So I don't think it's actually. I think it doesn't have a transmembrane, but it probably assembles with some other transmembrane protein. That's my guess. And we've expressed it. Yeah, I mean. Or, I don't, there's nothing like, there's no lipid moieties in there. So I don't think it's GPI linked or anything like that. But we are doing that in vitro now. And we're doing, or when we've done a lot of that already, really, where we're expressing these. There's no marine cell lines really. So we're doing mammals, but so far things seem to be doing what they would be predicted from their structure. So.
Okay. Well, Tony, thanks a lot, man.
Oh, thank you.
It was a great talk. Thank you very much. And I hope to see you soon physically.
Got it. All right,
here in Bogota.
Looking forward to coming down. Thank you.
Yeah, thanks so much.
Really appreciate it. See ya.
Thank you.